तर 5 मिनिटात सुरू करताय सर?
हो, अनाउन्स करून टाका.
हो हो हो, मुळे सर.
ठीक आहे, ओके.
बरोबर. हो, भट सरांना लावलं, तायडे सरांना लावलं का? डॉक्टर साहेबांना.
मॅडम नमस्कार.
नमस्कार सर.
नमस्कार. काय मॅडम कॉलेजमध्ये गेली, थोडीशी मीटिंग काय येईल म्हणे थोड्या वेळात. म्हटलं चला आपल्या लोकांना तितका भेटून घ्यावं आपण.
नमस्कार.
नमस्कार नमस्कार. अरे अरे हो दीपक, आवाज ऐकू आला पण दिसून नाही राहिला ना. व्हिडिओ चालू कर.
हे चालू नाही म्हणाला, हे काय म्हणते त्याला.
आता माणूस दिसला, अजून व्हिडिओ नाही चालू झाला.
आवाज येतो आहे ना?
आवाज येत आहे. व्हिडिओ का ना, आता दिसला. घरी आहे?
हो, घरूनच करतोय.
मी घरीच आहे. मॅडम गेले कॉलेजमध्ये, येतो म्हणे 5-10 मिनिटात. म्हटलं चला 4 लोकांची भेट होऊन जाते, आपल्याला काही काम नाही धंदा नाही. रिटायर्ड माणूस. तर मस्त आराम करून राहिलो. सर मला वाटतं ऑफिस मध्येच बसून आहे प्राचार्य साहेब.
हा सर.
ऑफिस दिसून राहिला.
याची तयारी सुरू आहे.
हो चांगला आहे. आमचं तर झालं मागे, आणि बी ग्रेड भेटला. नवीनच्या मानाने चांगला ग्रेड भेटला, कारण आमच्याकडे फक्त बीएससी आहे, बाकी काहीच नाही.
कोरोनाच्या याच्या लॉकडाऊन मध्ये लटकून गेलो, आमचा एसएसआर राहून गेला.
नाही आता होऊन जाईल म्हणा. आता येऊन राहिले.
पण झालं आणि पास पण झालं.
सबमिट झालं का?
एसएसआर सबमिट झालं.
एसएसआर अजून नाही केला.
ऍप्लिकेशन केली होती की लॉकडाऊन मुळे आम्ही सबमिट करू शकत नाही म्हणून. रिप्लाय
EQAR केले असतील ना सबमिट, आणि डॉक्युमेंट तुमचे त्या वेळेसच अपलोड झालेली असतील. प्रॉब्लेम येते सर, रिझल्टचा डाटा जेव्हा मागतात ते विद्यापीठाच्या शीट्स आपण जेव्हा पाठवतो, त्या स्कॅन करून टाकल्या की तो इतका मोठा होते की त्यांच्या ह्याच्यात बसत नाही, आणि ते म्हणते रिड्यूस करा. आम्ही रिड्यूस केला डाटा, तर ते काय वाचताच येत नव्हतं, इतका खराब झाला. आणि तसं झालं की मार्क्स कमी होऊन जाते आपले. ते दिसत नाही म्हणून मग ते कन्सिडर करत नाही. रिसर्च पेपर्स खूप जास्त होते, तर मग ते रिड्यूस डाटा केला की दिसत नाही म्हणून ते कन्सिडर नाही करत. आणि आमच्याच प्रॉब्लेम UGC केअर लिस्टचाही होता. केअर लिस्ट जी आहे ती 2-4 वर्षापासून सारखी बदलत आली. आमच्या खानचे काही पेपर्स होते त्याच्यात आता ते नंबर नाव दिसूनच नाही राहिलं. त्याच्यामुळे त्यावेळेस काही लोकांनी नंबर डाउनलोड, हा एक प्रॉब्लेम आहे. नवीन त्यात नाही दिसते दिसत.
डॉक्टर साहेब झाले?
बरं शशी भाऊ आले काय, वहिनी आल्या का?
येईल ना थोड्या वेळात येईल.
तिथूनच जॉईन होऊन जा म्हणा, वेळ होतो आहे कारण की समोरचा माणूस जर्मनीचा आहे.
तुम्ही चालू करा ना.
रवी
झुमडे मॅडम
सर, हो जुमडे सर,
सुरू करून द्या.
व्हिडिओ ऑन करा ना, व्हिडिओ ऑन करा सर तुमचा.
तुम्ही सुरू करा,
हा
काही प्रॉब्लेमच नाही. शी विल कम.
Good morning, everyone. I welcome all the dignitaries, all the participants who has admitted in our international e-webinar on recent trends in chemical and physical sciences on 20th September 2021 at NILKANTHRAO SHINDE Science and Arts College, Bhadrawati. So, dear
मॅडम
friends,
थोडं स्क्रीन खाली करा तुमचं. मॅडम हे दिसत नाही आहे. थोडस राईट.
so dear friends, chemistry is often called the central science as it plays a vital role in all aspects of physical and biological sciences, including engineering, agriculture, medicine, and aligned health disciplines. Department of Chemistry, NILKANTHRAO SHINDE Science and Arts College, Bhadrawati organizes international e-webinar on recent trends in chemical and physical sciences on 20th September 2021. So it gives me immense pleasure to extend to you a very warm welcome on the behalf of NILKANTHRAO SHINDE Science and Arts College, Bhadrawati, who has clubbed all of us for in the international e-webinar on recent trends in chemical and physical sciences. I welcome all the dignitaries and all the participants in this international e-webinar. So, friends, President of today's international e-webinar is Honorable Dr. Vivek N. Shinde sir, Management Counselor, Gondwana University, Gadchiroli, and President of Bhadrawati Shikshan Sanstha Bhadrawati. I welcome Dr. Vivek Shinde sir in this international e-webinar. Honorable
Recording in progress.
Honorable Chief Guest, Dr. Deepak Tayde sir, Associate Professor, Government, Vidarbha Institute of Science and Humanities, Amravati. Welcome sir in this webinar. Today's resource person, Honorable Dr. Ravindra P. Jumde sir, Research Scientist, Helmholtz Institute for Pharmaceutical Research, Germany. Welcome sir. Dr. Jyotindra Kumar J. Bhatt sir, Assistant Professor, Department of Chemistry, Kutch University, Gujarat. Welcome sir. Dr. Gajanan Mule sir, Head, Department of Physics, Sant Gadge Baba, Amravati University, Amravati. Welcome sir. Dr. Mrs. S.S. Aswale Madam, Associate Professor, Department of Chemistry, Lokmanya Tilak Mahavidyalaya Vani. Welcome madam. Dr. A.B. Wadekar sir, Assistant Professor, Department of Chemistry, DMB, Science and Arts College, Shega. Welcome sir. Dr. D.A. Poon sir, Assistant Professor, Head of Chemistry, Department of Chemistry, Shri R.R. Lahoti Science College, Morshi. Welcome sir. And organizer of this program who motivate us to organize such type of events is none of Honorable Dr. L.S. Ladke sir, Principal, NILKANTHRAO SHINDE Science and Arts College, Bhadrawati. I welcome sir to this international e-webinar. Welcome all the dignitaries and welcome all the participants who club at this international e-webinar. So I would like to invite Dr. K.P. Zumde Madam for introductory speech.
Very good morning, everyone. I am Dr. Curie, Assistant Professor of Chemistry, NILKANTHRAO, SHINDE Science and Arts College, Bhadrawati. It's a moment of great privilege and honor for me to extend an introductory talk. First of all, I heartily welcome for honorable, respected Dr. Vivek Shinde sir, President of Bhadrawati, Shikshan Sanstha Bhadrawati, Dr. Karthik Shinde sir, Secretary, and Dr. Vishal Shinde sir, Joint Secretary of BSSB, who provided us with such an ambitious place to pursue our goals and always make valuable, enthusiastic efforts for strengthening the institution. We all have gathered here today virtually to join an international e-webinar on recent trends in chemical and physical sciences organized by the Department of Chemistry, NILKANTHRAO Shinde Science College, Bhadrawati. It's a great honor for us that Dr. D.T. Taide sir, Associate Professor at Government Vidarbha Institute of Science and Humanities, Amravati, benefited as the chief guest of today's e-webinar, who is a well-known dignity and in the field of the education and research. In this COVID-19 pandemic situation, we can't come together, but our ideas can. So these digital platforms open the door to the researchers, professors, and related specialists to elaborate their ideas, their research work in their respective fields through the keynotes, oral presentation, or the poster presentation. These virtual conferences, workshops, webinars provide a distinctive pathway to share our ideas, research experiences, and participate in the interactive discussions. So, by considering this, the theme of our today's international e-webinar, recent trend in chemical and physical sciences, the resource persons of today's event contributes their valuable knowledge about the new research innovations. So here, for the opening keynote plenary lecture, Dr. Ravindra, postdoctoral research scientist from the Helmholtz Institute for Pharmaceutical Research, Saarland, Germany, will discuss about the opportunities of the research work abroad for the PG students and from his research experience journey of Italy, Netherlands, and Germany. He will also discuss his work on the drug discovery and approach to tackle the SARS-CoV-2 replication also. Our second resource person, Dr. Jyotindra Kumar Bhatt, sir, Department of Chemistry, Kutch University, Gujarat, will discuss the pragmatism in the drug discovery and development with the current post-COVID-19 trend, gives us an important ideas about the today's pandemic situation. Our third resource person, Dr. Gajanan Mule, sir, Head of the Department of Physics, Sant Gadge Baba University, Amravati. Amravati encourages us to enhance our knowledge about the nanotechnology from his plenary talk on the nanoparticles for the non-linear optical applications. In this way, the aim of today's international e-webinar is to stimulate our participants to explore the latest development and innovations, including the recent applications and assessment of the future commercial trends and opportunities in the science and technology. On this occasion, I specially extend my gratitude to our Honorable Principal, Dr. Ladke sir, organizer of the today's webinar, who always inspiring and encouraging us. At last, I gladly invite all the virtual attendees to bring your precious active support and participation, experience research, exploration, and invaluable achievement through our platform of the on the same trends in the chemical and physical sciences. Thank you. Thank you, ma'am.
Thank you, madam, for your introductory speech. Now, I welcome Dr. L.S. Ladke sir, Principal, NILKANTHRAO Shinde Science and Arts College, Bhadrawati, to deliver his inaugural address as well as report of the college. Dr. L.S. Ladke sir. Sir, सरांना unmute करा ना.
Audible?
हो येत आहे सर.
Very good morning to all of you. Respected chairperson and patron of this international webinar, Honorable Dr. Vivekan Shinde, President of Bhadrawati Shikshan Sanstha Bhadrawati. Dr. Vivekan Shinde, Secretary, and Dr. Vishal N. Shinde, Joint Secretary of Bhadrawati Shikshan Sanstha Bhadrawati. Chief guest, Dr. D.T. Taide, Associate Professor, Government Vidarbha Institute of Science and Humanities, Amravati, Maharashtra. Resource person, Dr. Ravindra P. Jumde, University Campus, Saarbrücken, Germany. Dr. Jyotindra Kumar J. Bhatt, Department of Chemistry, Kutch University, Gujarat. Dr. Gajanan Mule, Department of Physics, Sant Galge Baba, Amravati University, Amravati. Dr. Asole sir, ex-principal. Dr. Organizing Secretary of this webinar, e-webinar, Dr. Aparna Dhote Madam, Dr. Gajendra Bedre, and Kiran P. Jumde. Distinguished participants, professors, scholars, researchers, students, and entire staff of the college. At the outset, I would like to welcome you all in this international e-webinar on recent trends in chemical and physical sciences organized by Department of Chemistry of the college. I extend you all a very warm and happy welcome on behalf of Bhadrawati Shikshan Sanstha Bhadrawati and NILKANTHRAO Shinde Science and Arts College Bhadrawati. Being the head of the institution, I welcome resource person Honorable Dr. Ravindra P. Jumde, University Campus, Saarbrücken, Germany, with all humility at my command and extend sincere gratitude. I feel greatly honored while welcoming resource person Dr. Jyotindra Kumar J. Bhatt from Kutch University, Gujarat, and Honorable Dr. Mule, Department of Physics, Amravati University, Amravati. I also welcome Chief Guest Dr. D.T. Taide, Sir, Government Vidarbha Institute of Science and Humanities, Amravati. Friends, our institution, since its very inception, has made a notable contribution in the area of research. Up till now, faculty members of the institution published near about 700 research papers in national and international journals, and one patent and produced 15 PhD from our college center. Our college completed 3 cycles of the NAC and going to face 4th cycle in next month. The institution is 2 steps ahead in the academic program also. At present, the result of university examination are declared, and I am pleased and feel proud to inform that following the previous record of the meritorious students from the college, 5 students of the institution are in the merit list of Gondwana University, out of which 3 are the first merit in BSc, MSc Physics and MSc Botany, and remaining 2 are the 4th and 8th merit of BSc. College has organized number of national and international conferences, seminars, workshops physically, and webinars on different issues of science in this pandemic period. Today, Department of Chemistry organizes international e-webinar on recent trends in chemical and physical sciences to throw lights on recent research outcomes in the field of chemical and physical sciences. This e-webinar will enhance the level of excellence and rethink in orientation of research such that the scholars can think in different way with full potential. This webinar will provide a valuable opportunity for researchers, scholars, and students to share their knowledge. I am sure from this webinar, you will have fruitful and rewarding exchanges. I declared here this webinar is inaugurated, and once again, I would like to express my sincere thanks to the patrons of this webinar, resource person, distinguished participants, students, scholars, researchers, invited guests, and well-wishers of the institution for being present on this proud and delighted occasion. Thank you so much for your time with all of us here, and have a bright future and a very good and healthy day. Thank you all. Thank you very much. Okay, ma'am.
Thank you, sir. Now, I am very glad to say that in our international e-webinar, total registrations was done, 275 registrations was done, and from abroad also, some people are joining in our international e-webinar. Thank you, sir. On the behalf of our institution, I welcome our chief guest, Dr. Deepak Tadekar, Associate Professor of most renowned institution under the government of India. He is working towards connecting young minds to strive towards success and achievement. We are grateful to him for accepting our invitation and becoming a chief guest of today's event. So I would like to call upon Dr. Deepak Tadekar to address chief guest address to our students as well as our faculty. Dr. Deepak Tadekar.
Good morning. Good morning to honorable patron, all the executive members of this education society, all the faculties of NS College Bhadrawati. Specially thanks to principal of the college and Dr. Shashibhu Aswale also joined this e-webinar series. I, Deepak Tadekar, welcome all of you for this e-webinar, international e-webinar session. Dear friends, when we consider the pandemic situation, in pandemic situation, it is not possible to carry out the offline conferences, seminars, and webinars, as well as workshop. So to overcome this problem, the new trend come in the research, that is online conferences, online seminar, online webinars. The key person, that is principal of this college, is very enthusiastic, and he always give near about all facilities and encourage the faculties to give the platform to the budding researchers as well as the faculties. So Dr. Dhote madam, Dr. Kiran madam, and all the faculties of this chemistry department, as well as the department of physics involved to take such event, and here they are going to carry out the e-webinar on the recent trends in chemical and physical sciences. The recent trends which are occurring throughout the world to overcome the problems in the different pathogens, different types of medicine, different types of research, different types of research which is carried out will be explored by the honorable keynote speaker as well as plenary lecturers. Dr. Zumre is a well-known personality in the research. He is a young and enthusiastic researcher. So his knowledge will update the research as well as how the students will get or how the researcher will get facilities to carry out their research in abroad. This will be explained by honorable speaker, that is honorable Dr. Zumre. Which types of research is carried out, will be carried out in abroad. He gave such idea of that research. At the same time, Dr. Bhatt as well as Dr. Mule will explore their research work and how the new budding researcher will get breakthrough in their research will be explained by these researcher. This is a good activity of chemistry department. Really, I myself appreciate the efforts of chemistry department as well as efforts of the principal and the college. And at the same time, the executive member of this institute gives facility, gives this platform for their team to carry out such research event. And during the introductory speech, honorable principal said that there is a one
महाराष्ट्रातले प्रत्येक कॉलेजेस मधले आहेत युनिव्हर्सिटीचे प्रत्येक कॉलेजचे
honorable principal said that there is a one registered patent. This is a great achievement in the research. At the Taluka place, when the college publish one patent, it is an honor of college, it is an honor of society, as well as honor of that particular faculty and that ultimately department. At the same time, he also explored that there are 15 PhD awarded students from this institute. This is a great achievement, sir. And near about 700 to 900 papers were published in reputed international journals. This is a great achievement of the college. And this college will arrange such types of seminars, national level UGC sponsored, as well as different types of conferences, workshops were carried out by chemistry department. I know the efforts of Dhote man and his team and her team. So, without wasting time, I conclude my speech. I thankful to chemistry department, honorable principal, as well as executive member who give me this opportunity to explore my thoughts. Thank you and have a good day.
Thank you, sir. I welcome Honorable Dr. Vishal Shinde, sir, Vice Secretary, Joint Secretary of Bhadrawati Shikshan Sanstha Bhadrawati, who has joined in this international e-webinar now. So now it is my privilege to invite Honorable Dr. Vivek N. Shinde sir, President, Bhadrawati Shikshan Sanstha Bhadrawati, for presidential address.
Hello.
येत आहे सर आवाज.
Am I audible?
Yeah.
Yeah. Good morning to everyone. Respected Taide sir, today's keynote speaker, Dr. Zumde, Dr. Bhatt, Dr. Mune, our beloved principal, Dr. Ladke sir, all the faculties from chemistry department, all the dignitaries, all the teaching staff and fellow researchers and those students, those who have joined. On behalf of Bhadrawati Shikshan Sanstha Bhadrawati, I welcome you all in this pleasant morning. Today's e-webinar on recent trends in chemical and physical trends. As I am a person from a medical field, it's altogether a different field for myself. But still, I would like to elaborate on some things. As such, we are conducting these online webinars. As we all know, this is a great innovations in technology which brought revolutionary changes in exchanging knowledge, information, and it taught us a newer method of getting together. As somebody has done research in this field, we all know that has already somebody done research in this field, and today we are getting benefited by the same. Similarly, it's my suggestion to all the faculties and researchers, those who are in a research field, always try to pass on benefit of your research work and knowledge to the last person of the society. If a last person of the society is benefited from your research, then only it is a what we say, we can call it as a real research work which you have invented. Similarly, academicians and the teachers should also try to bring in newer technology, innovative methods in teaching, so that it will become easier to even a last student of the class to understand the subject, and it will become more easy and easy to him or her to understand the subject. See, being a head of the institution, it's our duty to provide you with all the facilities, all the what are the materialistic things which are needed, infrastructure which is needed to provide. But to use it at its maximum and to pass the benefit of all these things to the students it's a what I feel is the responsibility of the faculties. So, and I'm proud that all the faculties from all the departments of our institution, they are doing it so very well. So I think in the recent past, it's almost a fifth or sixth national conference which we are conducting with a good number of delegates. They are joining and taking benefit of it. One more thing I would like to say, what I think should try to promote students also to participate and be a part of such webinars, conferences, and research projects, so that they will be more and more. They can explore the things, and they will be well versed with this newer technology methods of getting together and of online webinars also. So I know that everybody is eager to hear the keynote address and the research fellows. So I congratulate the Department of Chemistry who has organized an international webinar of this on this topic. My best regards to all, and I'm pretty sure that you all will have a nice and deep discussion on today's topic. and definitely it will be beneficial to everyone. Once again, my best wishes to everyone. With this, I conclude. Thank you.
Thank you. Thank you very much, sir. Now, I am glad to invite Dr. Shahas ma'am to introduce the first speaker, Dr. Ravindra Zumre, sir.
Thank you, Aparna madam. Good morning, one and all. For President of the today's function, Dr. Vivek Shinde sir, Secretary of the Samstha, Dr. Karthik Shinde sir, Joint Secretary. Am I audible?
Oh, madam.
May I continue? May I continue?
Yeah, yeah.
Joint Secretary of the today's function, Dr. Vishal Shinde sir, Principal of the college, Dr. Ladke sir, and Chief Keynote Speaker of the today's function, and my Guru, Dr. Taide sir, Organizing Secretary and Head of the Department of the Chemistry, Dr. Aparna Dhote madam, Dr. Bendre sir, Dr. Kiran Zumre sir, speakers of the today's e-conference, Dr. Zumre sir, Dr. Bhatt sir, Dr. More sir, and chairperson of this session, Dr. Wadekar sir, Dr. Punda sir, all the dignitaries, all the delegates, and all respected participants. First of all, I would like to thank organizer, especially Dr. Ladke sir and Dr. Aparna madam for giving me the opportunity to chair this session. I feel privileged to welcome you all on behalf of the Department of Chemistry, NILKANTHRAO Shinde Science Arts College, Bhadrawati, to the International e-webinar on recent trends in chemical and physical sciences. Ladies and gentlemen, this is the first technical session. Lecture in this technical session will be delivered by young and enthusiastic personality, Dr. Ravindra R. Zumre. He is postdoctoral research scientist of Helmholtz Institute of Pharmacy and Research, Saarland, Saarbrücken, Germany. He completed his bachelor degree from Chandrapur, from Amravati University. He completed his master degree from Sant Gadge Baba Amravati University. He is awarded with the PhD from University of the Pisa, Italy. He has published 23 research papers with high impact factor. He has presented 16 research papers in various conferences and schools. Currently, he is co-supervisor of 2 research students. He has teaching experience of about the 15 years and research experience of about the 10 years. He received various postdoctoral grants under various schemes. The notable schemes under which he got the awards and the scholarship, the first was the Marie Curie postdoctoral grant, second one the senior postdoctoral grant from the University of Groningen, then Institute of the Molecular Science and Technologies, ISTM, National Research Council, CNR, Milan, Italy. Postdoctoral grant from University of Pisa. Another prestigious fellowship, it is given to him, that is Galileo Galilei PhD fellowship, University from University of the Pisa. He is an experienced and self-motivated researcher with expertise in the field of organic chemistry, polymer, supported catalysis, flow chemistry, and drug design and optimization, bringing forth a proven track record of successful and managing multidisciplinary research project and international research team. We are fortunate enough to have such a great personality with us. He will deliver a talk on dynamic combinational chemistry in drug discovery approach to tackle SARC-CoV2 replication. Now, I would like to hand over this lecture to him. Thank you, sir.
Thank you very much, Aswini Madam, for really nice and overwhelming introduction. Can I share my screen? I think the host is disabled.
Yes,
The
yes.
screen
Yes.
sharing. Yeah. Can someone allow me to share the screen because it's disabled now?
Aparna madam. Aparna madam.
हो मॅडम केलं को होस्ट केलं मॅडम
Ha.
Okay, that's good. I hope you can see my screen now.
येस 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 सर
Perfect.
So convert it into PowerPoint presentation. Huh?
Now I think it's in a full screen.
Yes.
Yeah, good. Good morning, everyone, and a special thanks to all the organizing committee for giving me this opportunity to present my work in this international webinar. So today I would like to present my research work on use of dynamic combinatorial chemistry in drug discovery. And I will give one example, how we find some compounds which can tackle the replication of coronavirus. And I think everyone knows now what coronavirus is, but I will go in details in my next slide. So just to give an overview, what I will cover in my presentation is my short personal journey. I think Madam explained it quite well. I will give you a little bit of more perspective on this field. Then I will discuss about research in European Union, especially about the budgets and which fields they are covering. Then I will give you introduction of drug discovery process and timeline, and the technique which I use, which is called target-directed dynamic combinatorial chemistry. Then I will jump into the results where we find some inhibitors of SARS-CoV-2, which is a coronavirus, using this technique. Finally, the conclusion outlook, and the last, I will show you which are the different research opportunities abroad for young and enthusiastic researcher from India. And finally, the acknowledgements. So starting my journey, I was born in Chandrapur. I did my PhD, I did my bachelor from Jagdamba Mahavidyalaya, and then I pursued my MSc in Shivaji Science College, Amravati University. After my master's, I went to Pune, and there I joined National Chemical Laboratory as a project assistant, where I worked for 3 years on different research projects, international and industrial and governmental research projects. And there I got an opportunity to know what are the different opportunities abroad for pursuing the PhD. And during that, I was able to apply for several grants, and one of the grant application was allowed or awarded to me, which was Galileo Galilei PhD fellowship at University of Pisa, where I work on asymmetric catalysis. And after completing my PhD, I worked in the same institute as an internal postdoc, and mainly on a nanoparticle synthesis. And then I moved to Milan, which is a northern part of Italy, where I continue my postdoc, and there I work on development of nanoparticle for catalysis purposes. Then after this experience in organocatalysis and metal catalysis, I wanted to pursue further in organometallic chemistry. And there I found a really nice group at University of Groningen, which also hosts a recent Nobel laureate, Ben Feringa. So there I pursued my second postdoc, and I work on development of new reaction methodologies, where we published several research articles in a highly ranked journals. Then after all this research work on chemistry, I always thought about what are their applications, like what are a real life application of them. And that brings me to my next application, which is a Marie Curie Fellowship, which is a highly prestigious fellowship in European Union and highly competitive. So where I wrote a proposal to show that how this methodologies, new methodologies which I developed can be applied in a drug design and development projects. So that I got a Marie Curie Fellowship in Germany, and in Germany I worked in Helmholtz Institute for Pharmaceutical Research, Saarland. This is situated on the border of France and Germany, and this is a really nice picture of my institute where I work, which is situated at the foothills of this nice forest. We have 3 floors. Each floor have a different expertise. So on the 3rd floor, we have a natural product isolation, and they're used as drugs. On the 2nd floor where I work, we are mainly medicinal chemists. We work on synthesis, development of new drugs, finding the new drug candidates and develop them towards the market or the preclinical studies. On the 1st floor, we have a drug delivery team. Here, whatever drugs we prepare, they are tested how to deliver them to different animals. So before going to the science, I would like to show how European Union funds research. So if you look at these balls, they are like the countries. So D is Germany, this is UK, this is Italy. This shows what are the average research and development investment from 2004 to 2009 and the GDP growth. So as you can see that the higher the R&D investment, better their GDP growth. So this is one of the things that most advanced countries, they tend to invest more and more money in research and development, and this reflects in their GDP growth. Unfortunately, in last decade, the share of European Union towards the R&D expenditure is falling down, and the China, Europe, and the United States are taking it over. So this is a good trend for the rest of the country, but for European Union, it was a kind of setback. So also, if you see the patent filed from EU, it was kind of now decreasing, and it was increasing from the United States and China. So European Union at the time thought, okay, this is not a good situation, and we should invest more money into the research. So although the patent and the funding was really going down, but the quality of the research was highest. So these are the different countries in European Union, and as you can see, they almost all of them reach their R&D target. R&D target is like when you write a project, you set some targets, right? And that targets are matched by these countries, which is remarkable, especially for the Finland, Sweden, Denmark, Austria, Germany. These are the countries where the most of the research and innovation is carried out. So same thing, the performance of these different countries, as you can see from the right-hand side of this bar chart, there is Sweden, Denmark, Finland, Netherlands, United Kingdom, which is in England, and these kind of countries, and also Germany, they are the innovation leaders. So most of the innovation in European Union are kind of driven by these countries. All the other countries are not bad. They are also above 50%, but you can see these are the leaders. So the Horizon 2020 was the European Union research objective. What they thought at the time that, okay, to boost this kind of research, what we should do is to get the integrated program, which covers the basic research and close to market innovation. That means that the research what we do in the university, it's kind of sometimes like really limited for a basic. But what European Union want at that time, that we do research which should be marketed, like the common people can get benefit from this kind of research. So the total budget was 77 billion euros, which was still less than 10% of the public R&D spending in European Union. So the European Union decided to fund these projects based on 3-pillar approach. What are the 3 pillars? I will discuss it in the next slide. So 3 pillars were excellence, industrial leadership, and social challenges. So they distributed the fund in these 3 different pillars. So excellence, this is kind of a basic research that is really essential to innovate. So ERC, which is European Research Council, so this is divided in these 4 different funding schemes. So ERC is for research fellows which are kind of starting now their independent research, like assistant professors also. So they get a starting grant of 1 million euros if they are able to successfully file this kind of application. Then there is a trend which is called future and emerging technology, which is kind of a basic research, but it's a little bit more in a technological field. So you get also funded for this kind of research. Then the Marie Curie Fellowship, which I got, it was also based on the basic research, but product driven, like which has some application. And some part of it is also divided in European research infrastructure. So you know that research is not possible if you don't have infrastructure, like if you don't have instrumentation. So European Union also gives fund for this kind of research. Then for industrial leadership, most of the fund goes to the key enabling technology. What are key enabling technologies? Key enabling technologies are like the technologies which are new and can be marketed easily. Then the risk and venture capital, which is like sometimes you open a startup company, and in the startup company you have more risk. But if succeeded, this kind of startup company can boost further and increase your GDP level. So in India also now we are getting a lot of fundings for this kind of ventures. So if someone wants to start their small companies, we are also funding quite well. Same thing goes in the innovation of KMUs. This is a key motive universities and units. Then the social challenges. So the fund is also going, like the highest fund goes on the social challenges. Social challenges means health issues. So to develop drugs for some diseases, food and security and safety. So also the research which is going in this field get funded quite well. Then the energy, what are the new resources for the energy, transport, climate change, society and security. So all these kind of fields get funded almost equally. So if you have a good research proposal, there is a high possibility that you get a fund to do the research and you get good infrastructure for that. Hope with that, I covered that which kind of research are kind of now going into the European Union in the different countries of European Union. And now I jump into the science. So science, I would talk about dynamic combinatorial chemistry and its use in drug discovery. So this is quite a recent technology. I will give you more details in the next slide. And then I will show how we were able to identify some compounds which stops the replication of coronavirus, which is called now SARS-CoV-2, which is severe acute respiratory syndrome, coronavirus 2. So before going to that, I think everyone should know how drug discovery process work and what is the timeline required for the drug discovery. Many times you might wonder that why we don't have drug for COVID now. And you will get answered by the end of this slide why we can't get drug immediately for such a disease. So the drug discovery process starts by biological target selection. So drug discovery process is not a process for one field. It's a combination of different fields which involves biologists, computational chemists, organic chemists, pharmacists, doctors, people, government, and all the agencies which you can imagine. So before starting to any drug discovery project, there is a first thing which we have to see is if there is a target known. What is a target? Target is a protein, or we can call it enzyme, which is responsible for function of that pathogen. Pathogens are like bacteria or viruses or parasites, right? So this biological target, the task is covered by bioinformatics, genomics, and genetic targets. So this is the field of biologists, which bring forward this kind of target. So you remember that in 2020, in January, when the COVID started, at the end of January, there was a biological sequence of coronavirus was published. So it was published by a Chinese researcher, and then whole research field, whole researchers in whole world use these targets with this genome and identify different targets and search for therapeutics for this kind of target. So this is the task of a biologist. Then there is a second process, which is hit compounds generation. So what is hit? I will come back to this point several times in my talk. Hit and lead compounds. So hit compounds are not a drug-like compounds. They are really small compounds. So this is a starting point for the chemist. So you synthesize several compounds, you test them on a biological target. If this kind of compounds inhibit or stop the process, biological process of this protein. So there are different techniques which are used to generate this kind of molecule. High throughput screening. High throughput screening means generation of thousands of compounds in parallel. Then computational screening. So there are several computational chemist groups working where you don't have to synthesize compound, but what you can do is virtually use these compounds and see if they are binding to this target, right? So every target, like every protein, has some cavity where these compounds can fit. And then it goes to initial synthesis. So once you find that, okay, some of these compounds which are virtually fit into the target, now we can synthesize them. And then assay development. What are assays? Assays are experiments. In a biological term, it's called assay. Assays are experiments where we check if the compounds which we synthesize, if they have a function, means if they can inhibit the function of a biological target, right? So high throughput screening at this stage, there are about 5,000 to 10,000 compounds synthesized, right? Synthesized or even purchased from some company, and they are tested in a biological target. Then out of this 5,000 to 10,000 compounds, there are lead compound generation. What is lead compound? Lead compound is like a drug-like compound. It's a bigger compound as compared to the hit compounds. And this compound has a definite activity towards the biological target. So here we started with about 10,000 compounds, and to getting to the lead compounds, we go around 250 or 200 compounds, right? And all of these compounds are kind of showing their activity against the biological target. So at this stage, there is a requirement of advanced chemical synthesis where the organic chemist comes into the picture. And then there is structure activity relationship, which we look for. What is the structure activity relationship? Structure activity is like if you have a compound and it has different substitutions on ortho, para, or meta position, and we see that only ortho position compound is showing some activity on a biological target, and meta and para position compound are not showing any activity. So this is called structure activity relationship. So if you change the structure, activity change. So this we look into, that's the lead compound generation stage. Then there are drug candidates. So after going from 10,000 compound to 200 compound, it goes to the drug candidate. Drug candidates where you are definitely know that this compound will inhibit your biological target, but then biological target in most of the cases is in the bacteria or virus, right? Just consider, just for example, look into coronavirus now. So coronavirus, if you kill it outside the cell, like outside the human, it's possible. You can do it by the disinfectant, right? But you can't use disinfectant inside your body. So what coronavirus does, coronavirus enter inside your cells, and we have to get our drugs inside our cells to kill the coronavirus, right? So at this stage of drug discovery, where the drug candidates are found, actually we develop a cellular disease model. What is cellular disease model? Is like we use cells from, suppose, animal, like for instance, rat or mouse or chimpanzees or human. You take that cells, you put it in a petri dish, then you infect that cells with pathogens like bacteria or viruses, and then we treat them with drugs or the compounds which we synthesize. And at this stage, the drug should enter into the cell without killing the cell. So that is most important part. And it should kill inside the bacteria or the pathogen inside the cells. So this is called cellular disease model. So here we are not injecting the drugs directly into the animal, but we are taking the cells out of the animal, we are infecting them with the pathogens, and then we are treating them with the drug. And here we also see about the drug selectivity. Is this drug selective for this kind of disease, right? So suppose I have now one cell which is infected by several pathogens, like influenza virus, COVID virus, and then there are some respiratory viruses. We infect them, and we see that if these drugs are selective towards the coronavirus, right, for example, and this drug selectivity is measured. So the compounds which shows a better cellular disease model activity and a drug selectivity are then carried forward in this process. So going from 10,000 compound to 200 compound, these drug candidates are around 4 or 5 compounds, maximum 5, not more than 5 compounds. Then there comes the preclinical development. Preclinical development, as I said before, it was a cellular model. Now this is an animal model. So what we do, for instance, in the rat or mouse, we infect them with, for instance, for example, just consider now the corona situation. We infect them with the corona. And then we try to use these 5 drug candidates and give them orally, intravenously, or intramuscularly, right? There are different pathways where we can administer this drug to this animal model. And then we see if the coronavirus inside the human or inside the animal is killed or not. And there we go for pharmacokinetic and pharmacodynamic studies. So for the bachelor and master student, I would like you to look into these 2 terminology, pharmacokinetics and pharmacodynamic. So what are they? So pharmacokinetic is kind of how your body reacts to the drug, like how it metabolize your drug. Pharmacodynamic is also like what is if you have a concentration effect of your drug. So if lower the concentration, lower the effect, higher the concentration, higher the effect inside the body. But to know it in more details, you should read it carefully. Then we also check if there is a toxicity of these compounds to the mouse model, suppose. So if we incubate our mouse, or if we inject this mouse with the drug candidates, and if it dies, then this is not a good drug. So we have to get it back, right? So then we also check a behavioral study, like what behavior these animals do. Like after giving this drug, if this animal behave really weirdly, so that is a no go. So if the animal behaves normally, then okay, we will carry forward the drug. If not, we will stop this drug and then go back to the synthesis again to synthesize different compounds and then go back. So this is kind of a cycle. So we do chemical synthesis, we get their structure activity relationship, and most of the time spent in this period is years. I will show you it in my next slide, that how many years required to go from the hit compound to the preclinical development. After you get a compound which shows really good activity in the animal, nice pharmacokinetic, pharmacodynamic, and the animal model is not even like animals are not behaving weirdly, then we carry forward these drugs in the clinical trials. So clinical trials, these are the human clinical trials. So there are 3 phases of it. Usually till here, we can do everything in academia, right? But to do the clinical trials, we required a huge fund, like billions of dollars, billions of euros. And here we have to somehow get industry into the action. So that's why we see like a lot of pharmaceutical industries, they are doing this kind of clinical trials. So these clinical trials are divided in phase 1 to 3, which are depending upon the number of people or number of patients which you put into the clinical trials. The phase 1, there are less people as compared to the phase 3. So when this already proven drug in the animal, we will put them into the human, so infected humans. For instance, if you have now corona, patients from corona, we put this drug into them and then see if they cure it. So this clinical trial also requires years. Then once this clinical stage is passed, that there are no adverse effect on the humans which were treated, then it goes to the government approval, government agency. So in US, you have FDA, Food and Drug Administration. In India, we have different agencies which do this kind of drug approval. So we have to go to them and we have to tell them, okay, we have this kind of study, and then it's called ICMR, yeah. So ICMR will then check into your study data and see if this drug is approved or not. And when it's approved, then it goes into the market. So after market, the whole drug discovery process is not end yet. So then there is a post-market safety surveillance. So which is like, you put this drug in the market, you can buy it from the pharmacy, and then you check like real-time uses of this. So suppose if the drug is now in the market and then many people are using it and now finding like most of them are getting sick or most of them are having some side effects, then many times this marketed drug is withdrawn from the market. There are several drugs, several examples of this that the people or the company spent like 10 years in developing this kind of drug, but after even marketing, we found that they are not good enough. So they have to brought them back from the market. Then I hope that I covered how the drug discovery process works. Now we should look into what is the timeline required for this kind of research. So the pre-discovery stage, which is like a biological target selection, can be done within months. That's not a problem. But then it comes to the drug discovery and preclinical trials, which requires around 3 to 6 years. So drug discovery, which contains hit, compound identification, lead generation, and drug candidates, right? These 3 steps, and then the preclinical trials in the animals, not humans, it requires 3 to 6 days. And as I told before, we start with around 5,000 to 10,000 compounds, like these are the hit compounds, then go to the lead compounds in preclinical trials, which are around 250. Then we go to phase 1, which is human trials. These are like 4 or 5 drugs candidate, maximum 5 drugs, which we can get. And then in the whole 3 phases of the clinical trials in human, it also requires 6 to 7 years. And after that, it goes to the review, to the government industry, and then large-scale synthesis and marketing. And here, only 1 drug can be approved. So starting from 10,000 compounds to 1 drug, which requires average of 10 years of research. That's why the question, whenever someone asks you why there is no drug for COVID, because COVID started now, 2 years before, there are companies which are working for it. Now, this period is now kind of reduced. So we will require maybe at least 5 to 6 years to get this kind of drugs in the market. So I hope that this covers how the drug discovery process works. And for many of you, this is kind of information which you never received before. But let's go to my institute, what we do in my institute. So I'm working, as I told you, in a department of drug development and optimization. So we focus on this part of the drug discovery process. So we have biologists in our floor, which selects the target. Then we have chemists which synthesize the compounds or check for the compounds and go from the hit compounds to the lead compounds to the drug candidate. For preclinical trials, they are not done at this institute, but we have a sister institute which are focused on this kind of preclinical trials. That's why I put it this box, half of the preclinical trials. But that is also in the academic setup. And this is like this first 3 to 6 years or 5 years, we do it in our institute. And what I am focused on is to identify these hit compounds and generate the lead compounds. So my goal is to find alternative strategies, so alternative ways to find these hit compounds and then optimize them to the lead. And the goal is to reduce this 3 to 6 year time to 1 or 2 years. So that is the most important part for faster drug development. And there I usually, my research lies on this area. So I told you about the hit compounds. So from where they come, like usually they come high throughput screening, like thousands of compounds are screened, or by the computational screening. But from where the basic idea comes from, like from where the structure, suppose if you have the structure of compound, from where it comes. So you will be surprised that most of it is come from the known compounds, so known drugs, right? So around 43% of this hit compound come from the known compound which shows some activity in a previous research. Then it goes to random high throughput screening, as so show here. So thousands of compounds are tested against the target, which is the protein. And then there are some direct approaches. I will not go in details about them, but the common function of all these different approaches, they are traditional ways. All the traditional ways, and they required more time to generate. And most of the time, the quality of these hit compounds are not that good. And then you have to go backward and forward into the drug discovery process to find the best compound. So as I told you, here I introduce my research, like how I reduce this time. So instead of going to this whole traditional ways, I will show you a new way which can generate a high quality hit compound. What is a high quality hit compound? High quality hit compound is the compound which shows a really nice activity, but it's on a target. And it has more chances to go through whole clinical phase, so whole process without failing. And here it comes the target directed dynamic combinatorial chemistry. So what is the unique of this chemistry? Here the biological target, I will discuss in details about what are the biological targets in my next slide. But just consider biological target is a protein inside a bacteria or virus, which is responsible for some of their function, right? For replication of their function or something like that. And here in this technique, we use this biological target. We throw some ligands inside that. And each biological target has some cavities where these compounds can bind, right? So what happened that this biological target, when we put some compounds, it kind of brings them together and the reaction occurs. So this biological target can also act as a catalyst for your reaction because it bringing two of the reactants together. And that's how it's forcing them to react. And when it reacts, it forms a new molecule. That molecule is fitting into the cavity of this biological target. And if it fits into the cavity of biological target, most probably that it will also inhibit, like stop the function of that biological target, right? So for example, so we use reversible reactions. So reversible reactions, this is acyl hydrazone formation reaction, which starts from aldehyde and hydrazide. So when they react it in an acidic pH, this reaction is reversible. So you will get always the reversible reaction when it is in an acidic condition. So for simplicity, how this approach works, what we do is so this head of these keys, right? These are the keys. So head of these keys, just consider these are aldehydes. And this tail of these T's are hydroxides, which are depicted in blue. So in one of the, in one vial, we throw all of them together. And when we throw all of them together, it tends to form different combination, right? So we have 2 aldehyde and 4 hydroxides, so they can form 8 different products, right? And that 8 different products and these reactants, they are always in equilibrium. And this is called thermodynamic equilibrium. So what we do, so whenever we throw external stimuli like a protein, right? Protein from, or the biological target, that's what I'm mentioning, protein into this library, this equilibrium shift towards right. How it shifts towards right? Because what is happening now? So suppose this second key head is fitting here, and then the second key point is fitting here. So what this protein is doing, so instead of choosing any other things, it's choosing these 2 components. They are bringing it together, and it's forcing them to form faster than the other products, right? So here is a nice example. So this is a protein target, and you can see there is, in the pink, there is this molecule. So this molecule fits into this cavity, and this cavity is responsible for one biological function of this target. This target was from a bacteria. So what happens now? Yeah. So as I told you that now this biological target force this compound to form over all the other compounds. And then what happened that you quench this reaction. How to quench reaction is to kill this protein, which is called protein denaturation. We kill this protein, we raise the pH to 10, like basic pH. In basic pH, this reaction is no more reversible. So whatever you form here, it's frozen now. So when it's frozen, that means that these compounds are final compounds and they will not go back to the aldehyde and hydroxide. And here, then you analyze this library. And in the library, you see that this compound is highly amplified or highly prepared over the other, right? It's 30% and all the others are 10%. So then we analyze it by HPLC-MS. So high pressure liquid chromatography, which is coupled with mass chromatography, right? Mass spectrum. So there we identify, okay, this compound looks that it's amplified a lot. So it's produced more than the other compound. That means that this compound is now fitting well to the target. So how do we perform this kind of reactions? They are really simple. They are micro scale reactions. So you work on around 100 micromolar concentration of this compound. So we set 2 reactions. So one is blank, where only, suppose, aldehydes and hydroxides, only the fragments are put into this kind of Eppendorf, which is called Eppendorf, this plastic tube, right? And in the other, we put these fragments plus protein, which is your target. And then when we analyze after the reaction, after the protein denaturation and then basifying the library, so freezing the equilibrium, we check and we compare the blank reaction with the protein templated one. And when we check the blank reaction with the protein templated, we see now these peaks belongs to products, right? So you can see number 1, number 2, and number 3, these peaks are higher as compared to the blank library. So what does it mean? This means that these 3 compounds were catalyzed by the protein. In other words, these 3 compounds are fitting well into this pocket or the cavity of this protein. And then instead of synthesizing now these 8 compounds, like in a large scale, we just synthesize these 3 compounds. And that reduce the synthetic efforts. So for instance, in a traditional way, what you will do, you will synthesize all 8 compounds, you test their biological activity, and then found out, okay, maybe none of them are active, right? In this case, you will synthesize only the compounds which are catalyzed by the protein, the target itself. And most of the time, these are the compounds which are really active. So what it does, it reduces the synthetic efforts in the process of finding drug candidates. So less synthetic efforts, better quality hit you are getting. And most probably that this hit will not fail in the drug discovery process. And at the final stage, it will shorten the period, which I showed, like this 3 to 6 years, right? It shorten this period of iterative cycle of design, synthesis, test, and analysis. This will reduce. So I hope that now I cover how this kind of technique is used to find the hit compounds or the good quality hit compounds. Hit compounds are the compounds which are used at the beginning of the drug discovery process. Now let's look at the example, right? So for example, I will show you, we use SARS-CoV-2, that is a coronavirus. And I think most of you know what is coronavirus by now. So coronavirus, now I was showing you, talking about biological targets and proteins, right? And I will show you now what are they, where they use. So with the example of coronavirus. So coronavirus has 4 structural proteins. What is structural proteins? They are responsible for its structure, the assembly of the virus. So first one is the spike protein. Most of you kind of heard about the spike protein on the coronavirus, right? This protein is responsible for formation of this kind of spikes on the coronavirus. And these spikes are responsible to enter into the host cell, like in our case, like human cells, right? Without this spike proteins, this virus can't be enter into the human cell. Then there are 2 proteins which are called envelope protein and membrane protein. So envelope and membrane protein are these 2 proteins which are responsible to form this spherical shape of this virus. So without this protein, this spherical shape will not form. So this is the function of this 2 proteins. And then there is a 3rd structural protein, which is nucleocaspid protein, which covers the whole RNA strand, right? The coronavirus is a plus sense RNA virus. Plus sense RNA virus means its RNA is coiled at the clockwise direction, and it has only one RNA. So this coiling and the structure of RNA is kind of governed by this nucleocaspid protein. So you know that when everyone says like, oh, wash your hands with soap to kill the coronavirus, what it exactly does? It exactly does this membrane and envelope protein. They are lipophilic proteins. And when we use soap, they kind of break this lipophilic protein. And then by breaking this lipophilic protein, this coronavirus is bursted and open, and that's why it's killed and it's not able to replicate further. So we talk about 4 structural proteins, right? And most of the time, you know, like all these vaccines which are now generated, they are for the spike proteins. So they are using this spike protein as a vaccine vector. So after this structural proteins, I will show you what are other non-structural proteins. So non-structural, in coronaviruses, there are 16 non-structural proteins, which are called NSP, non-structural proteins. Each of these proteins have a different function. And the most important part, that all these proteins are only expressed inside the host cell. So these proteins are not expressed when the virus is on like a surface or in the air. No, they are only expressed when the virus is inside the cell. What are their functions? So most of the time, the function of all these non-structural proteins is to transcript. Transcript means cut the RNA into fragments, and replication. Replication means to find these fragments and generate a new RNA virus, right? So all these non-structural proteins are responsible for transcription and replication of RNAs. And then when these transcripted viruses or transcripted RNA are like form a new RNA, then these non-structural proteins, these 4 structural proteins play their role. So they form this assembly. So this RNA is encoded now So this RNA was responsible, was formed by the non-structural proteins, and the structural proteins now encapsulate it, like cover it, and form this whole envelope, right? And this is how the replication of coronavirus is done. So I will not go into each of these non-structural protein and what their functions are, but I will show you some of them, like 2 of them. As I already told you, these non-structural proteins are expressed inside the host cells and very important for viral replication. So just remember it, the non-structural proteins are responsible for replications of virus, and the structural proteins are responsible either to enter into the host cell or the assembly of the coronavirus, right? And one of the most important points which I want to highlight here is that all these non-structural proteins are highly conserved. What is highly conserved means? So highly conserved throughout the different coronaviruses. Most of you might know that we have 4 different coronaviruses which are present around us and which are known as like common cold viruses, right? But they are not that pathogenic. They are not that dangerous like SARS-CoV-2. So there are these 4 coronaviruses, the old coronavirus 1 and this new coronavirus. In all these coronaviruses, these non-structural proteins are highly conserved, means they are not changing. So they have more than 90% of similarity. So there is less mutation going on or no mutation going on for this kind of non-structural proteins. On the other hand, if you look at the spike protein, you know now the Delta virus, which is revolving around the world, is now due to the mutation on the spike proteins. So if you try to develop a drug for a spike protein, most probably it will not work after a few years or even after a few months. So there are a lot of research groups which are also working on this kind of field, like developing a drug for the spike protein. But why we choose non-structural proteins? Because if we prepare a drug for a non-structural protein to inhibit its function, like if I now talking about the preparation of drug, that means I'm talking about preparation of chemicals which can inhibit, which can stop their function, which is transcription, like breaking of RNA, and replication of RNA. So if we kind of stop this function, we might have chance to develop a broad spectrum antiviral drug. What is broad spectrum antiviral drug means? That means that it can be effective not only for the SARS-CoV-2, that is the coronavirus 2, but it also responsible for the old coronaviruses or the common cold viruses. Most of the people knows that there is no cure for, or there is no drug for common cold, right? So this kind of a drug discovery approach and use of this kind of non-structural protein is really important. And I will talk about non-structural protein 14, number 14 and number 10. So this is the protein, and I will talk in details about what is their function. And now you will know what are the biological targets and proteins I'm talking about, right? So this is how the protein looks like. So in the green, this is NSP12. So NSP is non-structural protein, NSP12. And in the red, there is NSP10. So this is a complex of NSP10 and 14. So what is the function of NSP14? So NSP14 has 2 activities. So it has the first activity, which is N-terminal 3', 5' exoribonuclease activity. What does it mean? That to simplify its terms, like both these activities are the activities which are responsible for transcription and replication of virus, like to cut the virus into the pieces and to assemble this virus into a new RNA virus, right? Like just for the RNAs. So these activities are for transcription and replication of RNAs. And NSP14 is responsible for this kind of activity. But what is the role of NSP10 then, if the NSP14 is having this activity? Actually, NSP10 is act as a ligand for this NSP14. So without NSP10, there is no activity of NSP14, right? So they should be into the complex to get this activity done. And there was a recent study in 2020 which shows that they prepare coronavirus in lab, and they use a knockout mutant. Knockout mutants mean they perform a mutation that this coronavirus is lacking the NSP14 protein, right? And this coronavirus, which were lacking with the NSP14 protein, were not viable. Not viable means they are not able to replicate. They are not able to leave into the host cells. So that means that really NSP14, that shows, or that is the proof that NSP14 is really important for the virus assembly and replication and its survival. Then there is a recent study which is published in 2021, which shows that this activity, this NSP14, is not only responsible for the RNA cleavage and RNA formation, but it also responsible for reducing the host immune system, like reducing the host immune response. So due to this protein, when it is inside your body, coronavirus, and when this protein is expressed inside your body, it also kind of trick or change your immune system in a way that your immune system will not react against this coronavirus. So these are the different function of this NSP14 protein, and if we stop this function, most probably that the coronavirus will not be able to replicate. So then it comes to which of this NSP14 and NSP10 we select for the drug discovery projects, right? So most importantly, I told you that this kind of proteins can be generated by biologists, like they can be expressed, purified by the biologist. And most of the time, these proteins are not stable at room temperature. So we have to work really at a lower temperature and everything. So in this case, one of our colleagues in our institute is able to purify both of this protein, NSP14 and NSP10. Then the problem was NSP14 was not that stable, but NSP10 was. So we decided to use it. And there is another reason why we use it. So NSP10 is a small protein as compared to NSP14, which is really big, right? And the active site, or I can say the cavities, right? Cavities in this protein, which you can see here. There your chemical compound will bind. So these cavities are located only on the junction of this ligand and the protein, right? This protein interaction site. So most of these cavities in NSP10 are only responsible for formation of this complex. On the other hand, if we consider the whole NSP14 is a very big protein and it has several of this kind of cavities. And if we use that protein for the drug development project, most probably what will happen that your compound will bind to every active site or every cavity, right? But this is not the case for NSP10. So if we bind our compound to this cavity, it will not bind to the NSP14 complex. So this complex formation will not happen. So if you put your compound here, right? And the idea then is to prepare the drugs for NSP14, the compounds which binds to NSP10, sorry, and then the complex formation will not occur, and then the whole viral replication and transcription will not happen. So that's why we choose NSP10 over NSP14 for our experiments. So first thing is to check all the biological targets, all the biological proteins are most probably not compatible with your organic solvents, right? Fortunately, this acyl hydrazone formation reaction is really good that it can be performed in water with a really slight amount of DMSO, which is 5%, right? So what we check, that we have a random library. So we use 3 aldehydes. Unfortunately, I can't show you the exact structure because of the privacy law at my institute. But just to give you an idea that we use 3 aldehydes and 7 hydrazides, right? And then we add NSP10, which is our protein. And the solvents were PBS is the phosphate buffer, which is in the water, DMSO, and aniline was the catalyst for this reaction. Because at higher pH, this reaction is not reversible, right? That's what I told you. But when you add aniline, which converts this aldehyde to imine, and then imines are more reactive than the aldehydes towards this acyl hydrazone formation. So, and in that case, the reaction is still reversible. So that's the role of aniline here. And what we found out that for 48 hours in this reaction condition with this different reagents, and this protein NSP10 was stable. So it was not degraded, right? It was not decomposed. And we also saw there's amplification. Amplification means like amplification of some of the products. So in total, you will get from 7 aldehyde and 7 hydrazide, 21 acyl hydrazones, right? So out of this 21 acyl hydrazones, there were several acyl hydrazones which were amplified over the others. And I will show you in the next slide how they are amplified. So this is the LC-MS chromatogram, right? Which, yeah. So LC-MS chromatogram. So this is a blank reaction. Blank reaction, I told you how we perform the blank reaction, where only aldehyde and hydrazides were used. And this is a protein templated reaction, where aldehyde, hydrazides, and a protein is used. Then these reactions are run in parallel. They have exactly the same concentration of each reactant. And then after some time, we quench the reaction. We quench the reaction means we denaturate the protein, we kill the protein. And then we basify the reaction so that it's not reversible. So we are freezing the equilibrium now. And when we freeze the equilibrium at this stage, we found out that there are some peaks which are now shown with this green arrow, right? These green arrows. These proteins or these compounds were amplified. So you see that this peak at 12 minutes is higher than the peak in the blank reaction. Same goes for the other peaks, other green peaks. So this means that this compound is amplified, or it's catalyzed more by the protein. So on the other words, these compounds are binding to the active site of the protein, right? And to simplify this, I prepared this bar chart, and where higher the bar chart, higher the amplification, right? For instance, with this first compound, which is at 1.5 minutes, which was not even present or really slightly present in the blank reaction. But when we add a protein, it increases a lot as compared to the other compound. So that means that it's highly amplified compound. That means that it's strongly binding to the protein. And same goes for all the green, these 4 compounds, which were highlighted in green. They were amplified more than the other compounds. And then there are some 3 compounds which are amplified a little bit. They are amplified, their peak area is a little bit higher than the peak area of the compounds in the blank reaction. So I'm sorry that I can't show you the exact structure again, but just to let you know that these were the 4 compounds which we synthesized them, because instead of synthesizing all these 21 compounds, we just synthesize the compounds which are amplified, right? And these are now in the real chemistry reactions, like large scale reactions. We work on a gram scale. Before we work on a microgram scales, right? So we synthesize only this compound. In this way, we also reduce the amount of reagents we require. So we have less money invested in this project as compared to the traditional medicinal chemistry project. And most probably whatever hits which we are generating from here, or hit compounds which we are generating here, they will be active because they were selected by the protein target, right? So how do we see that if these compounds are really binding, right? There is a surface plasmon resonance study which we use to know the kinetic. So it is KD. KD is called there is dissociation coefficient. So lower the dissociation coefficient, better they are binding. So all these 6 compounds shows they were binding at a different level. So this compound, number 1, binds better as compared to the other. Then we also use if they are binding by the MS, so to form the complex of NSP10 and this product, and check by the MS if we observe the mass of this complex. And we found most of them were binding to the NSP10. Then what happened to the final coronavirus? So we have a collaboration with Professor Thomas Pichman from TwinCore. So TwinCore, where we have this virus lab, so viral lab, which is a safety level 3 lab. So where what they do, they use the cells from the animal, they infect it by coronavirus, right? And in this case, it was the old coronavirus, the human coronavirus 229E, which was easy to handle as compared to the new coronavirus. So they incubate, they infect these cells, and then they add our compounds to that, and then see if this coronavirus is killed. So these two graphs, which you can see, the lower the graph, the green one, which shows the infectivity of the coronavirus, which is reducing now. So this is a concentration of your compound. So once you increase the concentration of compound, the infectivity, the virus load is going down. And the other one is the cell viability. Like, you don't want to prepare any compound which kills your cells, right? So it's higher this value, better it is, and lower this value, better it is. So this shows that at least 2 of these compounds which were synthesized were able to kill the coronavirus in the cell. So, and now we are going into the SARS-CoV-2 assay. So this is the new assay now, which is developed in this institute, and we will test all our compounds on a real coronavirus now. So this brings me to the conclusions and outlook. So I hope that I show you that the target-directed dynamic combinatorial chemistry is applied well for this NSP10 target from the coronavirus 2. We also found the 4 binders for this protein, and using this approach, and 3 of them shows in vivo activity, so in cellular activity against this old coronavirus without any toxicity. And now we are also looking into the actual activity of this compound, and this compound will be tested on the new coronavirus, and then further optimization will carried out. So this is the end of my talk, but before going to the acknowledgement, I would like to show which are the opportunities for the young and enthusiastic researchers abroad if they want to pursue PhD or MS masters in abroad. So there are a lot of funding agencies which you can apply for. Yeah. So I would talk about each country. So for Germany, there is a DAD fellowship, which is a fellowship, and you can take a screenshot afterwards for this slide when I finish this slide, and you can go to the particular website and to look into the details. So that fellowship is for the master's and PhD students. So who has the capability to do the independent research, they can apply also for this DAT fellowship, which is basically from Germany. Then there is a Denmark government scholarship. Then there is also a France Eiffel scholarship, which is called, and it is also for the MSc and PhD. And this Denmark scholarship is also for bachelor's, master's, ME, and PhD. Then Holland is the Netherlands, where I also worked. So they also have a nice set of fundings for new researchers. Then there are also in Italy, for bachelor, master's, and PhD, you can look for this Italian government site. I will give you the site at the end of this slide. In Sweden, in Switzerland, and in Belgium, these are fully funded scholarships. They are really highly competitive. You need to be really scholar to get this kind of scholarship, which are bachelor, master's, and PhD. And then in UK, United Kingdom, there are this Bill Gates scholarship for the Cambridge and Rhodes Trust scholarship for the Oxford. And all this information you can find on this site. You can note it down. But before going to that, I would also like to tell you what they are looking for, right? Because not everyone can get this kind of fellowships. So what are the requirements? So the most important requirement, they need someone who has knowledge of research project. So who have high quality research project in MSc. So preferably from the well-known research institute or industry. So I would encourage for the master student, if it is possible, then try to do, some of you try to do your project, master's project into the industry or the institutes like National Chemical Laboratory, IICT Hyderabad, or there are some pharmaceutical companies where we are also promoting this kind of research project. So please go there and do the research, and then you get exposure to different instruments like in experience. So they also want people who has a basic knowledge of analytical instrument like GC, LCMS, NMR, this kind of thing. And you will get this kind of exposure in this kind of institutes, right? Then you should have a basic knowledge of literature search, how to do the literature search. That is important because they will check it during your interviews. They will check how do you write your proposal. Then publications are not required. Like you shouldn't have publication, but if you have them, like publication in international peer-reviewed journals, not into the conferences, right? So if you have publications in international journal, they are highly possible that you will get position. Then you should have a good future research project. So you have to write a future research project to get this kind of grant application. So you write a nice research project, they will evaluate, of course, your academic record. If you have a nice research project done before, if you have experience, and if you fit into all these kind of requirements, most probably there is a chance that you can get one of this kind of fellowship. So go through this site, and maybe you can find, if you have some problems, you can also contact me. I can guide you through how to do this process. Okay, this brings me to the final slide, which is acknowledgement. And I would like to acknowledge the people from my department, like from DIDA, Professor Anna Hirsch, who is the head of the department. Then Dr. Sebastian, who is doing this protein purification, whatever proteins I showed you. Then Sandra, she's a PhD student who is working on finding if these compounds are binding to the protein. Then at TwinCore, as I told you, they have a safety level 3 lab where these coronavirus assays or coronavirus experiments are running. And this is with the Thomas Pichman lab. And there are 3 students who are working on that. Then for another MS, neonatal MS, we have a group on the 3rd floor of Professor Rolf Muller and 2 of his co-workers. And they were also highly acknowledged for their help in this project. And fundings, yes, Helmholtz Association and Saarland University for funding this research. To fund my research, Marie Curie, I highly acknowledge the funds which European Union give me, and the European Research Council for funding all these research projects. Then I would like to also thank the organization committee and Dr. Vivek Shinde, President of Bhadrawati Section Society, Dr. Kartik Shinde, Dr. Vishal Shinde, who are Joint Secretary and Joint Secretary respectively, and Professor L. Ladkesa, the principal of NILKANTHRAO Shinde Science and Art College, for giving me this opportunity. Of course, the chief guest, Professor Dr. Tairesa, organizer, Professor Dr. Ladkesa, and whole organization committee, Dr. Aparna Madam, Dr. Gajendra Sir, and Dr. Kiran Madam, for organizing this webinar and giving me opportunity to work. And of course, all the virtual attendees for attending. I hope that you get some knowledge from this webinar. And thanks.
Really fantastic presentation, sir.
Thank you very much, ma'am.
You speak in detail about the funding in the field of the research, and also you have discussed the investment in the research throughout the world. This is very knowledgeable, sir. And you specially speak on the burning issues today. We are facing the problem of the COVID, and this is the important topic today. So you have focused on that topic, and this is really important for us and all the human beings. And you also discussed that the preparation of the drug is not the easy, it is a tedious job. And near about 5 to 6 years are required to prepare the drug for the COVID. But like layman, we can think that we want the drug for the COVID instantly, but it is not easy. You have already discussed in detail, and you have also guided our student for various fellowship and scholarship. So you are the source of inspiration for our young researchers, sir. Thank you very much, sir, for this stimulating and fantastic presentation and very knowledgeable presentation in this e-conference. On behalf of everyone and all the organizing committee, I would like to thank Dr. Jumde sir for nice presentation and sharing his idea with us. Thank you, sir.
Thank you, madam. And thank you, Jumde sir.
Thank you very much.
For valuable information. Thank you, sir. Thank you, Aswale madam, who accept our invitation and becoming chairperson of a technical session 1. Thank you, madam. Now, I am glad to invite Dr. Ajay Wadekar sir to introduce our second guest, Jyotindra Kumar J. Bhatt sir.
Thank you, ma'am. Welcome everyone in technical session 2 of the international e-webinar on a recent training in chemical and physical science organized by NILKANTHRAO SHINDE Science and Art College, Bhadrawati. I'm very much thankful to organizer team of our today's webinar for sharing this platform with me. First of all, I requesting to Dr. Jyotindra Kumar Jatashankar Bhatt, please occupy the chair as a resource person. Please, sir. I, Dr. A.B. Wadekar from Department of Chemistry, Sri Dhaneshwar Muskoji Brungle Science and Art College, Shegaon, welcome you in technical session of this webinar. It's my pleasure and honor to introduce resource person of this session, Dr. Jyotindra Kumar Jatashankar Bhatt. Currently, sir, working at Department of Chemistry, Kutch University, Bhuj, Kutch, Gujarat, India. He has completed their MSc and PhD. But sir have a huge teaching and research experience of 26 years. Sir paid their devotion. Sir paid their devotion as a good researcher with a multidisciplinary research interest in organic chemistry, medicinal chemistry, heterocyclic chemistry, analytical chemistry, green chemistry, and agricultural soil and water sustainability project. Sir has successfully completed 2 research projects and 1 project is going on. But sir work as a referee for PhD Gujarat, Sant Amravati University, Saurashtra University, Rajkot, Gujarat, and Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. Sir also work as a reviewer for 3 peer-reviewed journals. But sir publishes 4 books with a ISBN number on a different topics and 15 research articles published in various UGS care listed journals. He was presented more than 40 posters and papers in various seminars. Sir also worked for American Chemical Society, American Peptide Society, and International Society of Chemists and Biologists as a member. This is a brief introduction of Dr. Bhatt sir. So without taking so much time, I requesting to Dr. Bhatt sir, please deliver your talk. Please, sir.
Okay. Thank you very much. Dr. Ajay Wadekar for such a kind word for me. Actually, let me introduce myself in one sentence. I am a student of chemistry. That is my whole introduction. Now, I also know that we are running out of time. Whatever the time was given to me, I am going out of this one due to my very fantastic lecture given by Dr. Ravindra Jumde. Such a nice presentations and informative also. Whatever the time will permit me, I will try to conclude whatever I should do today. So let us try to have the presentations. Am I audible and are you getting the presentation?
Yes, sir, you are audible.
Yes, sir.
Okay, the presentation is going on, right? The first slide is there.
Yes, sir, very beautiful, sir. Yes, sir.
Okay, okay, sir. Okay, sir. So today, I would like to share with you about the pragmatism in drug discovery and development with a current post-COVID trend. This presentation, the topic that I have selected is for a general and a whole purpose to have a global condition of this COVID-19 pandemic. After that, we are gradually and slowly getting better worldwide. And our guest of honor, Professor Taide, said it was not easy to arrange the offline conferences, and still we are on a e-webinar. But it is a very good platform for all of us to share some ideas for the current trends and current demands of the world, our country, our institutions, and our society whole. So I would like to share something points which is in a general way, general trend of what we supposed to do and what is the situation. What is the drug discovery in a today's scenario? Aim and objectives of today's drug discovery trends of development work. What does that mean? I would like to throw some light on the current position of world, because once we have a pandemic, we are getting scared and we try to go for a betterment everywhere. So at some point, our research, our drug development programs, our research programs are having a very complexity. And this complexity is interfering a lot. During last years in 2020, our departmental research, our institutional research everywhere. So some point, it is a less pragmatism approach towards the development, and that affect a lot, and the span for the development of the drug is going to be increasing. So what supposed to do, how it can be worked, how we can come out with it, these things I would like to share. Our point is, until we really need for the alternate of this drug, we don't had a drug for a COVID-19. We were relying on only vaccine. So how drug research can get, and a lot of hurdles are there, a lot of complexities are there. So we should have to have some kind of a pragmatism inside in the research program, in a designing of the drugs like that. So demand is always at high, while the drug development and discovery, this is a DD and D, it is a drug discovery and development, isn't at the same level, at the same point, which demand is sitting over there. So demand is always high compared to our productions, our research findings. And the one point is, backfire in the development is also not avoidable, because sometimes we are getting failure, and this failure shouldn't keep up in a discouragement, but should we go to fight, and we have to also backfire to this failure, and one day we need to success. So at that time we require some time, so we can have some plus minus in our research. So we should have to have a pragmatism in our work. Let us say objectives are changed due to the global demands, and each and every country, and we can say India and rest of the world, need to change their objectives, their purpose, and their goal over. Overpopulated countries like India and China, the fast technocrat world, what we are running with, more comfort in our lifestyle, we already adopted before 2-3 decades ago. And healthy and nutritious food is right now in a crisis condition because of each and every human being who have a comfort lifestyle. They require nutritious food, and still we are having the lack of it. Some points we are going to be in a reducing source. So that should be change the global demand. Rarely freedom to multinational or national organizations for the explore their ideas directly in the development program, and due to the some kind of a like a FDA in US and everywhere, and we have our FDA, so the control is always there, and that's why at some point you can't able to explore your ideas, which is really need for the current conditions or situation. So this is one of the rare freedom is the most scenario. Universal fundamentals of drug development and discovery are immobile in positive way till the date, and we everybody know that global trends, it still need some way, some particular fundamental things on a piece of paper, which is we are saying MOU or something like that. So before that, if you add some kind of pragmatism, it should go in a mobile condition. So you will have a chance to reduce the time for a research and drug discovery. What companies are doing? Companies approach towards their specific demand, which is not global. Company obviously want to make money, and they have some kind of a specific target. But one more point I should say that company also came in front in this pandemic conditions, and they open their door for mankind also. But in general, I am talking, companies always try and focus on their specific demands. So it should be go in a global which has some kind of pragmatism. The gap need to bridge between parallel research institutions, universal drug discovery and development programs in worldwide. In our India also, we have some kind of a parallel research institute. One person are working over here in Gujarat, another person's working over in the Maharashtra or in Delhi or in Calcutta, in Bangalore. But if this kind of research is going on and nobody has a lack of communications or lack of maybe, I'm not saying awareness, but lack of communication is the proper word. So this parallel research is going on, and somebody has found a good idea, and while the another one have ideas, but they're not able to implement that one. So in this way, parallel research and the universal DD&D program should be bridged to remove this gap. That is the point. Boost this idea, think globally and act individually. This golden sentence should be adopted to get some kind of pragmatism in our drug discovery program. Now, a couple of questions I would like to share. This is the why, how, when, where, what. Why pragmatism is required? Because obviously for the human life endurance. And that is why each and every individual researcher should give this answer of this, because human life is, important. If there is no life, what is the meaning of this research? So human life is much more a counterpoint of this our endurance. How? With applying global power by the United Nations and World Health Organization, they can apply their power, and they can They can add one by one building block. Building block, I am saying one country is a one block. Our amino acids is a building block for the manufacturing of protein. Like say, each and every part of the world should be a one building block, and the power should be go with the US and WHO, so we can have some kind of pragmatism in drug discovery program. When we have to do like this, we are just keep talking about this one, each and every seminar and conference, no. But before it's become too late for mankind. Like this pandemic, we suffer a lot. And from this one, I mean, obviously we'll learn something about that. We are still keeping social distance, I mean physical distancing, wearing masks, something like that, wash oftenly our hands, we should not touch everywhere. These are the basic things we already learned from this condition. So before it's become a too bad conditions or situation for the mankind, we have to adopt pragmatism in our DD&D, where every country, I just told you about a one kind of building block as a one country in which they are having the comfortable eco-friendly zone and the talent. Talent is everywhere in each and every part of the world. We people are most talented, and we prove that we are scattering a different part of the world also. The intellectual people of the India, they are having a lot of they are doing a lot of very nice work in the rest of the world. So the comfortable eco-friendly zone is required, and talent should be adopted inside. On this particular place, you can develop DD&D. What we have to do? Executions of universal plan for drug discovery and development. We have to execute this plan by taking the help of United Nations and WHO. They can establish if the world is required. What type of factors that are influencing on PD over DD&D? The first of all, global marketing strategies. Hopefully it is complex because multinational company, national companies, they have their own R&D, they have research into scenario, focus, and very specific kind of research. And it is required. I'm not saying it is not like that, but this specific research also should be go in an expandable mood and expandable way. So we can have some kind of a less complexity in a marketing strategy. Cost-effective raw materials and the finished product ratio should be go in a rhythmic way. So common man can afford the medication which is required for their survival. Lack of universal legal perspective in overall drug discovery and the development. It is a D&D. It is a discovery and development. Lack of universal legal perspective in overall discovery and the development. So I mean, some countries like US, Europe, and some European countries with United States, they have some kind of a legal perspective. It should be go in a universal way. Like India, we have our own legal perspective, but by organizations like WHO, they can make a bridge for the legal perspective, and they can avoid this lake also. So all over world can have a nice drug discovery process, and not only discovery, but development process also. Low density of research tied up with medical centers. This is one of the major issues in our India, particularly. I am giving you one of the examples in the United States. Couple of universities. Everybody knows that in United States, industries, academic linkage is very strong, very broad. So our density is compared to other one is low, tie up with the medical center. So university researchers, professors, faculty members, PhD students, they should tie up with the medical centers. University should take initiative, as well as medical centers give the platform to the researchers. So this a low density can go in a high way. So something will be come out with a pragmatism approach. Next one is the applications of artificial intelligence. Still to implement. Right now, just my previous colleague, Dr. Ravindra has given some ideas, some highlights about all the techniques which are utilizing for the SARS-CoV-2 virus research. So this is one of the part. More software should be developed, more intelligence should be implemented inside of the DD&D. So it will be make a large platform, and it will make a frontier for the newcomers, new researchers also, and they will save their lot of time in their development. So artificial intelligence is, I think, one of the part to improve and to reduce the time of DD&D. Scattered colony of research institutions, universities, industries, and policymakers. Till the date, we are in a scattered colony. Institutional research are different, then the university research are different, industries are different, and policymakers are different. If they will come in a one platform, they will combine, they will share their ideas, they will strongly bond, and they will have some kind of better understanding. So this scatteredness will be gone, and the colony will become one of the society, which is a strong society, to get together DD&D in a very, very gentle way, I can say, and it will be benefit for the whole society and whole world also. Noteworthy changes in the environment. It is one of the factors that influencing, because right now a lot of crisis is going on. Somewhere is flood, somewhere is a lake of water. A lot of things are going on within a very short period of time. Before 2 centuries ago, it was not in a point out, but Brazil conference for the environmental science changed the whole scenario in the not only in the environment research, but in the drug discovery and design also. So environmental changes which is affect to the human life should be taken in a positive way and incorporate for a pragmatism in a DD&D. What steps should be taken for betterment and the resurgence? I think we should go through with the library for the research. Library, it is a very nice thing. What I'm not talking about the physical books library, I'm talking about the library which was the previous research. It is already going on, and at some point it was failure and it was put it as a library, like Dr. Ravindra said about the combinatorial way. So combinatorial chemistry is one of the best source to generate the library, and this library should be go for the research, re-research also. I should say re-research, because our search is only depend on the literature survey. It should not be like that. We should use the library and we should go through with the failure things. So that maybe the time will come, within short period of time will come, that we can have some kind of a good finding for a current kind of situation. Maybe the situation is different, but maybe you will have a more and more findings, and these findings will make you in a position to design the drugs in such a way that can be take a very less time period, very less factors that will affect on the development, and we can have a nice result also. Next one is the incorporations of a CAD, in silico, artificial intelligence, etc. AI, I told you. Now I would like to throw light on CAD, computer aided drug designing, which is the current requirement. Right now, we don't have a time to go through with the books and literatures and everything. We should have a one-click program to avoid the headaches of the time consumption, because we required this time. It is our precious time for the development. And that's why CAD and in silico both should be incorporated with some kind of pragmatism because once you go with the CAD, computer-aided drug designing, you require a very hi-fi software also. But to go through with this one, you should tie up with the other institutions, and that's why in the beginning I told you the gap should be bridged. Again, my next point, this gap also should be bridged with the global scattered R&D institutions. So you can incorporate CAD in silico and AI, and you can have a better resistance. Academia, industries, global NGOs, and the government should be a module for the DD&D under the control of World Health Organization. This is a demand of current world. And we understood last years, in 2020 years of our very hectic years it was. And each and every people of our, not only our country, but whole the world were getting scared about what will happen tomorrow. So at that time, some institutions should take initiative. And who is the best source, I think, to keep everybody get together? Reduction in time consuming process. Obviously, it is oftenly I am telling time is a most precious things than platinum and gold. Support to green and eco-friendly nanotechnology. Nanotechnology is right now going on with the drug delivery system. It should be penetrated in the designing of the drug and the development of the drug also. And green approach is there. We are running with the green chemistry and eco-friendly catalysts we are utilizing right now. But it is the, if we talk in general, it is maybe 10%. I am getting scared to say 10% also. I'm saying high one. But we should increase the green and eco-friendly with nanotechnology support. And it will be give a better resurgence. Encouragement in a failure and repeat model. This is a very important thing. Just go through with the library for research and it will give some kind of encouragement. If students will failure, we are giving encouragement. This is not a one time. You can go, you can try to next time, you will be better. So this things is repeat model should be incorporated for a with a, some kind of pragmatism which is required. Let's see what world researchers are saying. I am just having a little less time, so I am just giving idea that this is the 2-3 reference I have taken. Despite of the advantage, in the drug research development, our knowledge of the underlying molecules, mechanisms of our many diseases remain inadequate. This have led to limited effective medicines for survivor diseases. To address these challenges, efficient strategies, novel technologies, and policies are urgently needed. The main obstacle in drug discovery and development are the mounting cost, risk, time frame needed to develop for a new medicine. And fair pricing and accessibility is another global challenge. So this challenge is still today remaining. We should try to avoid these things also. Global vaccine R&D effort is response to COVID-19 pandemic is unprecedented in terms of scale and speed. Everybody knows, friends, we know that how long it takes. But think about the previous Ebola vaccines came after such a long period of time. And right now, due to the some kind of a little positiveness, globally acting with the help of different institutions research, we can able to reduce time factor and we had vaccine. So this scenario should be keep continue with some kind of pragmatism. Industries benchmark for traditional vaccine developed pragmatism cites additional rates for licensed vaccines more than 90%. So we should think about it. Finally, the strong international coordinations and cooperation between vaccine developers, regulators, policymakers, fundraisers, fund givers, public health bodies. Government will need to ensure the promising late-stage vaccine candidates can be manufactured in sufficient quantity and equivalently supplied to affected areas, particularly low resources region. We should think this globally and act individually, because we people, Indian, we are dense populated and with a very late facilities towards the common people. So this little pragmatism, increasing in pragmatism of a DD&D, and we have a good number of different kind of vaccines for a different kind of diseases. Maybe another diseases, who knows what will happen tomorrow. So before that, we try to adopt this pragmatism in our DD&D program, so we can avoid some kind of this situation and we can fight better. Pandemic had lent a new urgency to debates that have been rumbling for years among the legal aspects and the civil society, pushing regulatory bodies to make over policy changes. COVID-19 pandemic remained a serious systemic mistake, and we all allowed to happen over the decade. A drug research and development systems is publicly funded, majority high-risk based research, but sells the need without condition and privatize the pods without hesitation, only for public be same drug back again in a export prices. So this thing is a very well known now, and that's why we should think about some kind of a positiveness, some kind of a relaxation in the discovery program. It has been widely acknowledged. One of the Dutch prime minister was in 2016 was saying that the system is broken. Patent and the intellectual property exclusives are only counterstone for the current model. And that's why some very focused, very aimed and with a very perfect objective we required to develop. And that's why if you want to do like this, you should have some kind of freedom. And this freedom can come with some kind of a pragmatism. This is a model. I should just give idea. Normally, new chemical entities from a basic research 10 to 25,000 compounds we can synthesize. No way. It is very easy to synthesize. But the second phase, which is a development phase, and having a 3 sub phases, phase 1, 2, and 3, everybody knows about this model. So how it can be go? You can see how the molecules is going to be reduced. And in the third phase, which is the 4th phase of our clinical trial and post-marketing surveillance, introductions of this unit, maybe only one guy, only one candidate can hit and can become a lead molecule. So this is, you can see, number of years is, and at least 12 years is already passed. Then only you will have at least one unique molecule candidate, which will go in a phase 4 for a post-marketing surveillance, will become a medicine. So to drug to medicines, the journey is a very, very long one. Each and everybody knows. And that's why to sort out this journey, to design drugs with some kind of a pragmatism, it is a need for today, along with the discovery, this DD&D model. So this, we are coming from the design to drug in the market. We know that at least 12 years of the span is taken to get the medicine. Now, just some highlights on the development of the 5 stage. Discovery and development, then the preclinical research is going on, then clinical development is going on. FDA will review and review again, and then give the approval. And then post-market monitoring will be happen. After this monitoring, if drug is reliable and non-toxic and everything is there, then it go to the market. So this drug development portions have a 5 stage, and this stage we need to understand in such way, and we know this 5 stage we have to go through it. But if you want to reduce the time factor, we should have to have some kind of a pragmatism. This Nature is published one article in October 20 about the way. So I am just going, it is about this, our COVID-19 pending situation. It's also saying that how the probable achievement therapeutic doses in the patient include this can be done. And it is also used. So how research and search for the current condition that is happening, and they published this one. This is the model, and we like this model to have the high throughput reframe and repositioning. Vaccine is the final that we know, but this is a model, and this model is already research model, previous library. That my point is to adopt, to penetrate some person that never needed is the best.
Am I audible, sir?
Yes, ma'am. Hello.
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 What, sir? Please continue, sir.
Yes, Dr. Vadekar, now I am audible. I think
Yes.
at some point the internet connection was loose.
Yeah.
Okay, sir. You are audible now.
Okay.
Please continue, sir.
Okay. So I am going to.
Yes, sir. Yes.
Okay. Getting the slide now?
Yes, sir. Yes, sir.
Okay. So this was the model. This is given by Laura Level in an article. So these things I would like to go with the investigational drugs approach. This is a very good understanding to go through with the previous research and library and then go ahead. So it will reduce the time. It will save the money. It is a cost effective also. So two things is there. Directly we can have benefit from this one. Now I am just giving a little highlights about our university, our research group milestone. I had started UGC funded minor research project during 1994 to 1999. And then I worked as a postdoc fellow in the reputed institutions. And then I migrate US for the postdoctoral research for further work. What we are doing at right now, I visited North Carolina at Chapel Hill during 2000 for my postdoctoral work. At that time, we try to establish the different peptide research work. I joined polypeptide laboratories at Toledo, California in the United States as an R&D chemist and worked for 6 years. And I was a scientist for the development for the peptide growth hormones release factors, GNR, gadolinium also there. We developed the leuprolide, which is right now utilizing for the prostate cancers, and octreotide, somatostatin, number of peptides, custom, generic, all these things were there. I was doing some kind of a development work in the peptide for a solid phase and solution phase both, with a Merrifield and different resins. The steps were synthesis, cyclization, peptide modifications is there. After following the purification by lyophilizing, and the isolation is there. So this was the strategy. This was the one of the production units. You can make a kilo in the terms of peptide with a very, very specific design. Right now, as a group leader, I am working with along my other faculties and research scholars and collaborative institutions like other university and industries. We are jointly working. Principal investigations role to keep alive with the students, academic institutions with a MOU, faculty members and affiliated institutions. Our work is in progress, is a development of new chemical entity with a littered design, like 4-oxoquinazolines as anti-cancer molecules, 2-amino-4-thiazolidinones as anti-HIV agents and anti-tubercular agents, peptide cap 2-azitidinone, which is the beta-lactam rings, which is constituted by the penicillin kind of broad spectrum antibiotics. So 2 peptide cap azitidine is with a very markable interest for our group. Substituted thiazolidinol as a potential SCARCO2 in silico studies, and the biological evolutions are on boulevard, and we got some promising results. Right now I cannot share, but this is a very interesting project going on. Synthesis of derivatives with a green approach, which is focused on a moiety and under development work with a green catalyst, micro-assisted synthesis. We try to develop green catalysts which can be utilized with a various field also. What kind of institutions we have with a collaborative work? Chemistry department of Kutch University with Solaris Chemical Industries, Agrocell Industries. We are working for the soil fertility, increasing the soil fertility for a different crop for the farmer. Department of Earth and Environmental Science, we tie up with this one for biological evolutions and DNA extractions like that. Center for Excellence in Chemical Sciences it is run by the Department of Chemistry. We are having the sophisticated instrument facility like FTIR, NMR, MAS, LCMS, GCMS, HPLC, UPLC, NMR, and SAM electron microscope facilities we have. Agricultural soil testing for the health card Yojana, which is implemented by the Prime Minister of India. Before it was introduced as a Gujarat state, then Mr. Narendra Modi was the Chief Minister, and they introduced this one. Our university, our department is working for them as a government project. This is our TRIMP. We applied for the 2 patents with a collaborative 2 universities. We publish research papers, articles with international and national as well as state level journals also in a conference proceeding. He is Dr. Malai. He is working as an assistant professor in a physical chemist, and he filed 1 patent. He is Dr. Murugesh Trivedi. He is an environmental toxicologist, consultant scientist, and we established a soil fertility laboratory also. Peptide research laboratory is also there. Division of Medicinal Synthetic Heterocyclic Chemistry, and this 3 laboratory is going on right now. This is our Center for Excellence in Chemical Sciences. And peptide research laboratory is upgraded, is in a pipeline. And custom synthesis and consultative projects are also in a track. Number of research works projects are there in the pipeline. We are having a slow motion, and due to this some kind of establishment. Who can do this one? Let me tell you about this one. What I should not tell about myself, I should tell who can do it. The students who are really enthusiastic for their career, they can do it. Academicians who wish to fashion their R&D work, they can do it. Every individual personality who has ability to think out of the box. Like we people living in India with a very lack of facility, but we can think globally. The day will come that we will have a nice result. So out of box thinking is a very important way. Well, as Indians, for our healthy society and wellness, we have to do like this. We have to adopt pragmatisms in our DD&D. If anybody wishes to get information about the abroad research study, please feel free to email me, because right now we are running with a lack of time. So I am just giving my email address. Anybody can contact me anytime. This is a very renowned, our president, former president of India. Don't read success stories. You will only get a message. Read failure stories. You will get some ideas to get success. And I like this one because DD&D can go in this way only. Thank you very much. I would like to thank NILKANTHRAO Shinde Science and Arts College, Bhadrawati, Chandrapur, for inviting me and give me this opportunity. I particularly, I would like to extend my sincere gratitude to Dr. Vivek Shinde, the president of this society, Dr. Ladke, the principal of the college, Dr. Aparna Dhote. She did a lot of headache for me. She coordinating every time with me for this opportunity. Dr. KP Jumbe, also from the Department of Chemistry. Dr. I would like to extended my sincere thanks to Professor Deepak Taide, who is the guest today, special guest of this webinar, who approached me to deliver this speech. And also previous my colleague, Dr. Ravindra, you give a nice, fantastic speech. I reduced my time because of your nice speech. I would like to also thank each and every participant who get maybe give your speeches time, and you may, I'm not sure, but you may get some kind of a positiveness and positive way of the thinking for DD&D. Lastly, I would like to thank you all whose name is not there with my that. If I forget somebody, so I am thank you as a special thank you everyone.
Thank you very much, sir. Your presentation is very nice. So this topic is now open for discussion from participant. If you have any question, sir, with us for the interaction.
Continue.
Okay. So I express my sincere thanks toward Dr. Bhatt sir for spending your valuable time with us along with your fruitful knowledge in drug discovery and development. This is outstanding presentation on very hot and crucial topic in this COVID pandemic. Sir explained so many important points under pragmatism in drug discovery and development with current post-COVID-19 trend. He started his talk with a drug discovery and development today and explored in detail about factor influencing on drug discovery and development, drug design and development model, and so on. Thank you very much, sir. I'm sure this talk definitely open new door of drug discovery and development for researcher. My special thanks goes to Dr. Ladke sir, Dr. Aparna Dhote ma'am, Dr. Bedre sir, Dr. Kiran Zumde ma'am, and my guru Dr. Diti Taide sir for offering me this opportunity. Lastly, I congratulate to NILKANTHRAO Shinde Science and Art College, Bhadrawati and organizer team for a grand success of this international e-webinar. And hand over mic to Dhote madam. Ma'am, please. Thank you.
Thank you, sir. Thank you, Wadekar, sir.
Thank you.
Thank you, Bhatt, sir, for your valuable guidance to our students, faculties. Thank you, sir. Now, first, I welcome Dr. Gajanan Mule, sir, Head Department of Physics, Santa Gadge Baba University, Amravati, who joined international e-webinar on recent trends in chemical and physical science. Now, I am glad to invite Dr. Dinesh Poon, sir, to introduce Dr. Mule, sir.
Good afternoon to all of you, honorable delegates, research person, and participants for today's international webinar. I request Dr. Gajanan Mule, sir, to occupy the chair as a resource person for today's international webinar for the third technical session. Dr. Gajanan Mule, sir.
Yes, sir.
Sir, excuse me, Poon, sir.
हाँ मॅडम
म्हणा शेअरिंग स्टॉप करा म्हणा त्यांचा मुळे सरांचं
काय
होणार
म्हटलं
भट सरांना सांगा की त्यांचं शेअरिंग स्टॉप करा म्हणा भट सर
I would like to
But sir, stop your sharing, sir. Stop sharing. Continue, sir.
Okay. It's my privilege to introduce Dr. Gajanan Mule, sir, on the recent trends in chemical and physical sciences, that is RTCPS 2021, for the third technical session. Now, the Gajanan Mule, sir, has completed his MSc from Amravati University by getting the first rank in the university. He has PhD from Amravati University in 2009 on the topic, the growth and characterization of lesser material and non-linear crystals. He has qualified NET GRF in June 2002, who stood in the 20% toppers conducted by UGC. And he also qualified NET for the lecturership in December 2001, SET in February 2002, DRDO SET, which is of scientist B grade, conducted by DRDO New Delhi in 2002, GEST JRF with percentile score 86.1248, conducted by IIA Bangalore. He has got the awards and honors from the Amravati University, Utkrishta Seva Gaurav Puraskar, Vidyapit Shikshak, Sant Gadge Baba Amravati Vidyapit Amravati, वर्ष 2017-18. He has also got the Excellent Article Award for 2018 for the research article Effect of Sodium Metasilicate on Structural, Optical, Dielectric and Mechanical Properties of ADP Crystals, published in Journals of Material Science and Technology in 2016. He is also the advisory committee member for National Conference on Upcoming Trends in Chemical Sciences, that is UTCS 2013, organized by Department of Chemistry, Sri Anand College of Science, Pathardi District, Ahmednagar, during 6 and 7 September 2013. He has got the second rank Best Poster Presentation, UGC sponsored National Seminar on Chemiphysics, February 28, 2010, organized by Department of Chemistry and Material Science, Sri R.R. Lauti Science College, Morsi, District, Amravati. He has got the first merit in MSc. He has visited South Korea to present paper in international conference and visited laboratories. He has a Senate member.
Continue, sir. Mule, sir.
Okay, ma'am. So, very good afternoon to all the participants, organizing committee members, and resource persons of this esteemed international e-webinar on research in chemical and physical sciences, organized by NILKANTHRAO, SHINDE Science and Arts College, Bhadrawati, District, Chandrapur. So, in this talk, I will talk on the topic nanoparticles for nonlinear optical applications. And the outline of my talk is somewhat like this. Firstly, I will give you just brief introduction about nonlinear optical effects, as this is the new topic. Then the thermal lensing effect and the nonlinear optical effect. Then I will briefly introduce the technique used to study the nonlinear optical effect, and then I will just make you be aware about our results regarding the whatever the studies we have made on the nonlinear optical studies in view of their applications in nonlinear optical devices. So we know the optics. Generally, we say the optics in which we study the how the light behaves when it interacts with the material, and in linear optics. So I have to, now we have to make a division that what is the linear optics and non-linear optics now, because I am talking about the non-linear optics. So here linear optics means the optics in which the properties of the materials, optical properties of the materials, such as the refractive index and absorption coefficient, it is independent of the intensity, and the frequency of the input light, it is not changes as it passes through the medium, so it remains same. So light cannot interact with light. So, for example, if you pass two beams from your medium, so the medium cannot affect, cannot have the medium cannot have any effect on that light. Okay, so in this case, we cannot say that the light is controlling light in the case of linear optics. And in this one, the polarization, which is governing all the optical properties of the material, it is remain linearly proportional to our electric field of light or electromagnetic waves which are passing through the medium. But in nonlinear optics, certainly the polarization will not remain linear with the electric field, and it becomes non-linear function of the electric field. Means it will depends on the electric field E square of the electric field, cube of the electric field, and so on. In this case, refractive index and the absorption coefficient, it changes with the intensity of the light. In this case, the frequency of the light passing through the medium, it is changes with the intensity. It is changes at the output. In this again, we can say light can control light. So the photon in the medium, it is interacting. So photon in the medium, they are interacting. So light controls the light when it is interacting with the medium. So that the light interact with medium, light interact with the light ion medium, and the presence of an optical field modifies the properties of the medium, which turns into modified the other signal. Okay. So in order to see the linear, non-linear optical effects, we shall require the electric field of that light. It is of the order of 10 raised to power 5 to 10 raised to power 9 volt per centimeter. So which is able to disturb or modify the interatomic fields, which is of the order of 10 raised to power 7 to 10 raised to power volt, 10 raised to power 10 volt per centimeter. But in linear optics, the field is very weak. Field of the light is very weak, we consider. It is only 10 raised to power, it is of the order of 10 raised to power 3 volt per centimeter, and which is not able to disturb the at the interatomic field at a large extent, so that we get only linear effects. The first, after invention of the lasers in 1960, first non-linear optical effect, it was discovered by the scientist Franklin and his co-worker in 1961. It is the second harmonic generation. So we have used ruby laser, and they focus this ruby laser on a quartz plate, and then they dispersed the light, and they found that the ruby laser has the wavelength 6943 angstrom, and they found that in addition to 6943 angstrom, the another wavelength at the output of this quartz crystal, it is 34715 angstrom. So this wavelength is again present. So which is just half of this fundamental wavelength, that is original wavelength, 6943 angstrom. So they confirm that there is a conversion, there is a doubling of the frequency. It is half wavelength means frequency is double. So in this case, the frequency is double. So it is said to be second harmonic generation. And it is discovered in 1961, and later on so many non-linear optical effects have been discovered. And now from that one, the trend has been set to discover new non-linear optical materials which have good non-linear optical properties, non-linear optical coefficients. So I'm not taking a lot of mathematics, but in order to understand what is the polar, what is really the non-linear optics. So firstly, we can consider the polarization. If you consider the electric field, this electric field, it is E is proportional to cos of omega t, where we can consider omega is the frequency of that electromagnetic signal or light incident on the material. Then polarization, it will develop in the medium, and we can have the polarization terms. This one P0, it is this DC term. P1 cos omega t, it is depending on the frequency omega t, which is same as that of the incident frequency. So we say it is a fundamental term. Then we can have the polarization at the output twice polarization in the medium of frequency twice omega, which leads to the generation of second harmonic or the second harmonic waves. Another way we have the polarization depending on the frequency 3 omega, means it is oscillating with the frequency 3 omega, so which is related to third harmonic term. So this way we can have, if the intensity of the radiation is very large, then it can set the polarization with the DC oscillating frequency omega to omega and so on. So that
तोच एक region आहे म्हणे का?
So that it will again emit the polarization, the oscillating polarization will emit radiations, and you will get these radiations of frequency omega 2 omega 3 omega at the output. Okay, so there are so many non-linear optical phenomena involved in this non-linear optics. So we are just concentrating on this second order and third order. So this second order phenomena, which are this frequency conversion, second harmonic generation, parametric amplification, parametric oscillations. Third order non-linear optical phenomena, those are third harmonic generation, self-phase modulation, self-focusing and defocusing, which is related to nonlinear refraction, stimulated billowing scattering, stimulated Raman scattering, optical soliton, and optical bistability. So these are the third order optical phenomena, and those are taking place if the intensity of the light is very large. So if you consider only the third order nonlinear optical processes, which is related with the third term in the nonlinear equation for the polarization, and we can consider this is one. So P3, it is third order nonlinear polarization. This is epsilon 0 chi3 E of T cube. So it is proportional to cube of the electric field of that light. And this chi3, it is third order nonlinear optical susceptibility. If you consider this E is equals to E cos omega t, and if you put it in this equation, so you will get these two terms. So one term it will consist of 3 omega, consisting of 3 omega, which is third harmonic generation, means it is oscillating with frequency 3 omega, so it will emit radiation of frequency 3 omega, and another term which has frequency omega, so consisting of cos of omega t means frequency omega. So this first term, it is related with third harmonic generation, and the second term, it is related with nonlinear contribution to the refractive index experienced by the wave at frequency omega. Okay, so this is the intensity dependent contribution to the change in refractive index. It is said to be a nonlinear refractive index term. It is occurring at a frequency omega, and it is depending on the intensity, and it is third order nonlinear optical phenomena. So if you consider the light, incident light or intense light, it is inciding on your medium, and it is a nonlinear medium, and as intensity is large, so we can write the refractive index n is equals to n0 plus n2r. N0 is the DC refractive index. We can consider it as a constant one, and n2r is the intensity of the light, and n2 is a nonlinear refractive index, and we can write this nonlinear refractive index, this one. So this intensity dependent refractive index, it produces or it shows so many interesting phenomena, and they have a lot of applications in this modern technology. So this intensity dependent refractive index, it shows this interesting phenomena, that is self-focusing and defocusing. So if you consider the Gaussian intensity profile beam, we are allowing to incident on this nonlinear optical media. So this laser beam, if it is a self-focusing, so it will converge. It will focus. If it is a self-defocusing, so it will diverge. Okay. So this way, this material modifies this beam if the beam has a high intensity. Another is optical Kerr effect. Optical Kerr effect is used in the switching of the optical switching in the different communication systems. So this optical Kerr effect, which depends on the intensity of the light. Because we are dealing with the higher intensity radiation, laser radiation, so there are non-local effect, which is there because of the increase in the heat, because of the high energy of the optical pulses or optical intensity. So there is a thermal gradient developed in the medium, and which leads to the change in refractive index, nonlinear refractive index. So those are said to be a non-local effect. Other interesting phenomena takes place if your light is intense one. So there is a saturation. So the change in the refractive index is caused by the nonlinear absorption of optical energy by the medium. So there will be a nonlinear absorption of optical energy by the medium, and which leads to the saturation, different kinds of saturations. So this self-focusing phenomena is used to
Due to some technical problem, we will start within 2 minutes. कोण सर?
हा मॅडम.
त्यांच्याकडे लाईट गेली आहेत सरांकडे.
बरं, मग
सर म्हणाले
कन्क्लुडिंग करायचं.
हा तर नाही तर सर म्हणाले 2-3 मिनिट वाट बघतो म्हणाले. तर थोडं सांगून द्या सरांना, म्हणजे अनाउन्स करून टाका. कोण सर?
Yes, madam.
Sir, conclude the session.
Okay. I would like to present my sincere thanks to Dr. Gajan Mule sir, who has delivered his nice information on the invited talk on nanoparticles for the non-linear optical application in very simple and language. He has shared the beautiful views on nanoparticles. Due to some unavoidable circumstances, he has concluded his lecture. So I again say thanks to him on the behalf of the committee. I would like to thanks all the organizing committee of NILKANTHRAO, SHINDE Science and Arts College, Bhadrawati. Once again, thank you. And I give mic to Dr. Dhote madam. Over to Dhote madam. Thank you, madam.
Thank you, sir. Due to some technical problem, Dr. Mule sir is unable to share his views with us. Sorry for inconvenience. Thank you, Phun sir. Now, thanking is the great tradition of every function that is organized in any form. It is proper end to give celebration where the dignitaries are thanked for their presence to the function. So I invite Dr. Jyotindra Kumar sir to propose vote of thanks.
Thank you, madam. honorable dignitaries, different professors from different colleges, and dear participants. This is the last part of today's e-webinar. First of all, I express my sincere thanks to the patrons of this e-webinar, Dr. Vivek N. Shinde sir, President Bhadrawati Shikshan Sanstha Bhadrawati, Dr. Karthikeyan Shinde, Secretary Bhadrawati Shikshan Sanstha Bhadrawati, and Dr. Vishal N. Shinde, Joint Secretary Bhadrawati Shikshan Sanstha Bhadrawati, for their valuable guidance and guidance for this organizing this webinar. Again, my special thanks to chief guest of today's e-webinar, Dr. D.T. Taide sir, Associate Professor, Government Vidarbha Institute of Science and Humanities, Amravati, who always helps us and guide us. Dr. Dhote madam always contact with Dr. Taide sir for their guidance, and we are very much thankful to Dr. Taide sir for organizing this today's webinar and guiding us. Again, I am very much thankful to today's speaker, Dr. Ravindra P. Jumde sir, research scientist, Helmholtz Institute for Pharmaceutical Research, Germany, for their today's talks and valuable guidance in today's corona pandemic. And again, I am thankful to Dr. Jyotindra Kumar, Assistant Professor, Department of Chemistry, Kachchai University, Gujarat, for their valuable guidance. Again, I am thankful to Dr. Gajanan Mule sir, Head Department of Physics, SGB Amravati University, for their nice informative lecture on nanoparticles. And again, I am thankful to today's chairman, today's chairman of Technical Session 1, Dr. Mrs. S.S. Aswale Madam, Associate Professor, Department of Chemistry, Lokmanya Tilak Mahavidyalaya, Vani. Again, I am also very much thankful to the chairman of second technical session, Dr. A.B. Wadekar, Assistant Professor, Department of Chemistry, DMB, Science and Arts College, Shega. Again, I am thankful to the chairman of third technical session, Dr. D.A. Poon sir, Assistant Professor, Department of Chemistry, Shri R.R. Lahoti Science College, Morchi. I am very much thankful to our principal, Dr. L.S. Ladke sir, for their valuable guidance and guidance for organizing this today's e-webinar. I am again thankful to Dr. Mrs. Aparna Dhote madam, Dr. Kiran P. Jumde madam, the organizing secretary of this e-webinar, and Dr. Mrs. Aparna B. Dhote, who conducted this program very well. Again, I am thankful to Professor Sandeep Pradhan sir, Kadri sir, and all the teaching and non-teaching staff of our college, and all the participants who joined this e-webinar. And I'm also thankful to all those who directly or indirectly help us in organizing this e-webinar. Over to Dr. Dhote madam.
Thank you. Thank you each and everyone for joining us with in international e-webinar on recent trends in chemical and physical sciences. Thank you, sir. Feedback link will be sent within 5 minutes in a group. Sir,
Recording stopped.